genome. So we know where they are located, but we now sort of want to figure out where are they located on the genome, and that's basically after mapping. And then there is a lot of quality measurements you can do on both this mapping step and also on like by examining where the reads are mapped based on your a priori assumptions where they should end up. The things that we're going to talk about and have work on is the mapping statistics that you get out from the aligner, and it could be star or it could be HiSat, and all of them are leaving some kind of log on how well they manage to map the reads. And then once they are done, you can, by using gene annotations, so basically asking questions like, are the reads mapped to the exons as I think, or are they mapped in other parts of the genome? Do we find the correct intron-exon junctions as we expect, or do we find a lot of other stuff? And other stuff like that is things that we can ask by using annotation and the mapped reads. Just to give you some things that you can get from the mapping logs is almost all programs are giving things like uniquely mapping reads, which is basically that the read that you submitted will only mapped to one place in the genome. So it's quite certain that it's in the right location. But then there are also multi-mapping reads, and that is reads that the program can't decide where it comes from. So basically, if you have a read that has a sequence that is found in multiple places in the genome, the program can't decide where to put it, and then it becomes a multi-mapped read. And then there are some reads that it just can't find at all, and those will be unmapped. And then there are some programs, they also give informations about mismatches and indels and how many splice junctions. And one of those programs that give all of this information is the STAR program. So this is basically how the log looks if you would go in and have a look at it. Yeah. So here you can see the speed by which the programs are operating in the human genome. So in 1 hour, you will get 211 million reads mapped. So it goes really fast. And here you can see how many of the reads that were uniquely mapped to the genome. And this is a really good number. This is sort of the things that you are hoping to get, but you don't necessarily have to, but that's fine. Then STAR is also giving the information about the number of spliced reads, and both the annotated and also reads that were spliced in non-canonical sites. So if you can look at the numbers, so now I'm trying to find my pointer, but yeah, here. So this is the number of annotated splice sites. So these different ones that are known to be in there. And these are the non-canonical, so the ones that are not known. So you can see that they are mapping much more preferentially to this one than this one is just most likely just noise. And then you get how many that have been multi-mapped here, and how many that, and the reason why they were mapped, and that's just in this case because it was too short. There was just no place where they could get all these seeds mapped. Yeah. Yeah, and you have for HiSat, the program that you're using, you don't get that much information. You basically get how many that were uniquely mapped, and that's when they are concordantly mapped one time. So in this case, 78% were mapped perfectly. And then, yeah, and this is the multi-map number. This one here. Oops. Do you see my arrow pointer when I'm, or not? Can I get a nod if?
Yes.
Yes, good.
I can't.
It's very small, but yes.
Yeah, exactly. I don't see it myself sometimes, so yeah. And yeah, and here you get the overall alignment rates. Now, what can you do with this kind of information? Well, this is one example where I was using this information, and that was when we were trying to determine which program to use for allele-specific expression analysis. And then we tested 3 different programs, STAMP, STAR, and TopHat, and you could see how many reads that were properly paired to the genome. And the thing that we were interested in was in a hybrid between two wild-type species, Grandiflora and Rubella, and we had created a hybrid that was something in between. Thank you, Roy. And and the hybrid then has, of course, one chromosome from Grandiflora and one from the Rubella. And the expression should be somewhere should come from both of them. The problem is that the reference, the annotation, was from Rubella, so the reads were mapping much better to that reference than the other reference. And this was mostly obvious when you were looking at the TopHat, where when you were not mapping to the correct reference, you had a much lower mapping ability, 65%, compared to when you were mapping the reads to the correct reference. So, and when you did a hybrid, it was something in between, of course, because half of them belonged to this one and half of them belonged to that one. But you could not see that same big difference when you were using STAR or STAMPy. So, and STAR was the one that was behaving the best. So by using just these log values, we decided to continue to work with STAR and left the other ones without too much concern. Yeah. So if you get a lot of bad mapping, so basically if you get 50%, 40%, you should at least spend some time to try to understand why that is. Is there something wrong with the genome that you supplied? Is there something wrong with the settings you did? Could it be that there's something else in some other species that you haven't thought of? Or is there just a lot of bad reads that are there because of a bad run or something? So you should spend some time on trying to understand it. But even though it might turn out that only 40% of the reads are mapping to your genome, that doesn't necessarily mean that you can't use those 40% and analyze it. It's just really important to keep it in mind that there might be something strange going on here. Okay. But now let's say you actually manage to get all your reads mapped to a good location and the statistics look good. So then you want to know where they are located. And then you can use this package, which is called RSeQC. And that program provides information that is very similar to the information that you get from FastQC, which are the ones up here. The difference is that when you did it the previous run, you were doing it on all the reads, whereas this information is now only on the reads that actually mapped to the genome. So there's a small difference here in which reads that are being represented. And then there are a lot of RNA-specific QC that you can't get from FastQC, and those are these things like have you saturated your samples? I mean, have you sequenced deep enough? and you can get answer for that. You can get the information about redistribution, coverage, uniformity, and strand specificity. And you're going to try out these programs. In general, this is how the different, so the program is of course different depending on what of the functions you are calling, but you always supply a reference, and that should be in this bed12 format. And then you supply your reads in the BAM file. So you need the reference and you need the BAM file, and then you give them where you want to write all the information. Okay, so this is one example of the files that you can get for RNA-specific information. And this is sort of answering the questions, are your reads distributed as you think they are? And in general, you can ask the question, are the reads degraded or not? And this is mostly useful when you're working with poly-A-enriched samples. If you have not degraded sample, it should look something like this. And if you have degraded sample, it's going to look something like this. Also, if you use 10x data or something like that, which are just sequencing the 3' end of the read, you're going to see a very similar pattern like this. And the reason why you see this pattern is because of, I'm going to try to explain it to you here. So let's say you have a pool of RNA, some with poly-A tails and some without, and you do your extraction with poly-A RNAs. And then you will only have those left. And then you convert them into cDNA and you fragment them into small pieces. And then you will have representation of the entire transcripts. And then you send that for sequencing, and then it means that you will have reads that are covering the entire gene. Let's say instead that you have a sample that has been degraded, so there's lots of nicks in the RNAs. They are no longer full-length RNAs. And then when you extract the ones with the poly-A RNA, you will get the poly-A and you will get only this small part that is sort of belonging to that one. All the rest of them are going to be lost in the sample, which means that you're only going to get the 3' end part of your read. And then you're converting that into cDNA and you're turning that into sequencing and you send it to sequencing machine and they are sequencing it. So that means that you're sort of going to get an enrichment of the 3' end of the reads, and then you will get this kind of curve. So when you're doing RNA analysis, if all your samples are looking like this, it doesn't necessarily have to be that bad that they are degraded. The big problem comes when half of the samples are looking like this and half of the samples are looking like this. Because then you are going to get different representations of the number of reads per gene. Because in this sample, even if you have a super long transcript, the representation of it is only going to be a short one. Whereas if you have like this, the super long transcript is going to have multiple fragments. Whereas in the degraded, it's only going to have a few. So then when you're doing differential expression, that is going to be a problem for the analysis. So where are the reads mapping? So if you're done poly-A, or if you think that most of them you hope that most of them end up in the exons of your transcripts. That you can do by using this read distribution. And then you will get information about how many ended up in the exons, how many ended up in the 5' UTR and the 3' UTR, in the introns, and also in the and before the transcription starts, right? So just upstream of the gene or downstream of the gene. And the one that is the interesting one is this tags per kb, where you can see that for the coding sequence and the UTRs, you have hundreds per kb, whereas in the introns and in the upstream and downstream of the transcripts, you have fives, ones in the 1 to 10 size. So it's a really big enrichment in the mature parts of the transcripts. So that's really comforting if you can see that. If you see that it's much closer to each other, then you have to think about, is there something wrong with my samples, or how should I treat this? Another thing that you can also have a look at is this kind of junction saturation, and this gives you information about. So this is on the left, and that one is giving you 3 lines. Where the blue line is all the junction that is being found, the red one is the known junction, and the green one is the novel junction. And here it says the % of total reads. So let's say, for example, that this sample had 100 million reads to start with. Then already after approximately 10 million reads or something, 15, all of the known junctions in the samples has already been found. So you won't find any more of the already known annotated intron exon junctions after 50 million reads. You have basically saturated all those. But then there are a lot of novel junctions, and those are new ones being found all the time. The more you sequence. And I think it's quite representable from this one. I don't know if you remember this from yesterday, where they were showing this example, like if you have multiple intron exon junctions, most of the ones that are being it's just, even though there are multiple junctions, it's only 1 or 2 or 3 of them that are actually being expressed at high levels. And then these ones that are coming up here are the rare ones, and probably noise. So if I would have had this sample, I would have said that, okay, we have spent too much money sequencing too deep. We are not really getting much more novel information that is usable after this level here. So this is typically a plot where I would say that you spend too much time sequencing one sample. And I would have suggested that you would have sequenced 2 samples instead at half the depth. If you have paired and reads, so I don't know if you have paired and reads, if you remember this slide, you have the fragment, which is the RNA that is in the cDNA, and you have paired and reads. So you have 1 read coming from this side and 1 read coming from that side. And what this one is telling you information about is the inner distance between these 2 reads. So this is sort of giving you the information about the distribution of the inserts in the fragments. And for this specific example, the insert distance, which is the one that is here, is on average 100. So I would say that this is quite a good distribution for you to have. And then you can see that there are some samples here which is lower than 0. And that means that the read 1 and the read 2, yeah, read 2 and the read 1 is reading into one another. So actually the inner distance is shorter. So that's why this can be a negative value. So here you can, so for these ones, it would have been better if the distance was bigger, because now a part of read 1 and read 2 are giving the same information. That's what you can see here. Yeah, so just this is sort of, so even though you see a lot of yeah, so things that doesn't make sense or things that wasn't like you expected, it doesn't mean that you should throw away your samples at this point, but you should really mark them up and keep that in mind when you start doing analysis downstream, that if you see changes that might be due to this, then you should keep that in mind. Again, a multi-QC can get all the information from RSeQC and put that in the same HTML file where you also have your FastQC. So once you've done all the RSeQC steps, run multi-QC as well, and then you will be able to add that information, so you have everything in one file. Yes. Are there any questions now about this?
I would have one. So I wonder, since you are working for the facility, how often will you get bad quality RNA samples, like degraded ones and so on? Because they're like 15 years, well, okay, 10 years over. People's experiences.
So it really depends on often it depends on where the samples are coming from. So for example, often when you're working with clinical samples or things like that, there's a big risk that the samples are not so good. Just because that wasn't the priority when they maybe did the surgery to remove the cancer or whatever. So then there's that's one time, and then there are all these where people are collecting samples from nature, and again it's sort of a problem to make sure that everything is put on ice and everything is perfect for making sure that the RNAs are not degraded. But I think now, I mean, so the facilities has been around for a long time. So I think the reason for the samples are being bad are getting less and less due to the sequencing facilities. It's more that the groups have had problems to get the samples in a way so it doesn't, the RNAs are not being degraded.
So do you mean like in the library construction, or that the facilities are better at constructing libraries? Or?
Yes. So in many cases, yes. I mean, in some cases, biology is strange. There are some species and some things where you have had to be working with the species for a long time to do good RNA extractions and stuff. But in most cases, I would say the facilities are doing a great job to extract RNA. That's my feeling. Any more questions?
Yes. Yes, I have a question. In the figures you showed about, the percentage of total reads and splicing, why you mentioned you think is not necessary? Because in my idea, it's just basically you will have a very nice, basically coming up with novel junctions, and also the all junctions have the same kind of trend. Why is here you just basically, I didn't get exactly what was the actually the sum up a conclusion why we just think is not okay.
no, it's not wrong. It's not wrong. It's just like it's. I mean, this is so there. There is this question: should you sequence deep or should you sequence a lot of samples? And in general, what we suggest is that you should sequence a lot of samples because it's better to have multiple samples to adjust for biological diversity. So basically, to be able to have statistical tools and enough statistical power to identify things that are differentially expressed. And the more samples you have, the better the results, or the more things you can identify. And yeah, and the other thing then you can do, of course, is to sequence deep to find new things. So and for me, and this is just my point of view, is that when you start to sequence deep, like in this case, when you get above 20 or 25, I think that the most of the new things that you find there are not functionally relevant anyway. They're basically just there because of the imperfection of the biological system that you're studying, and that's and I think that holds true for 99% of the cases. Then there are, of course, all these 1% cases where you read about fantastic, amazing biological findings that are super cool. So I'm just saying that if you want to find these things, that these 1% things that are super interesting and groundbreaking, then you have to be, then you would have to have like other, you would have to have a good plan on how are you going to filter out the ones that are interesting. Did that answer your question?
Yes, absolutely. But that was very nice, actually. How many do you think is a good number of replication, biological replication in that kind of case? If you want to be more confident, depends on that kind of reads.
Yeah.
What do you think? Your idea, I want.
Yeah. So again, I think that you can find things that are differentially expressed, or I think you can find junctions that are going to be different between two settings and still not functionally interesting. But in general, I think the less likely something is to occur, the more samples you would need. And the more diverse things that you're comparing, the more samples you're going to need. I think, are we going to discuss this more in detail at some other point, or should I take it now?
We might talk about it a little bit later, but yeah. I mean, just in summarizing basically what Johan also said, and also on a vision that we share usually, is that RNA-seq will not prove that you have that, for example, that the splicing junction is very important for your data set. You might find many relevant findings, but in any sequencing data, you need also to validate in another method to prove that that is actually happening. Because most of the finds that you can find here might not be actually functional, as Johan is mentioning. So that what he's actually focusing is that you need some other ways to validate your data to prove that that is a really biological finding rather than a technological artifact or analysis artifact in that case.
can I add one point? Sorry to basically get the time, but yes, I agree. We need the functional analysis or behavioral studies or something else. We confirm actually this is going to be effect. I agree with you. But the point is based on the statistics and based on the basically the power of this analysis, how we can just basically be sure how many biological samples enough to just basically I stand and I say, okay, in later on I will confirm it with just functional analysis or some kind of behavioral studies or other things to confirm the behavior or the behavior of these genes or that function. But I need a statistically standing for it based on this analysis, and I say, okay, I think there is a highly possible this is the place is just functional.
Yeah.
Yeah, that will actually depend a lot on which kind of question do you have. So for example, if you have patient samples, then hundreds. If you have biological replicates, so which is the case of this course, so 3 replicates is enough, or maybe 6, depending on which kind of material you have. So yeah, so that power analysis is really data set dependent, I think, to get like this really what is actually the importance of that gene.
Thank you, because I need some kind of ideas, and that is great. I got my answer. Patient 100 and also basically between 3 to 60 replicate. You are happy as experts. That's great. Thank you very much.
And we have also a question from Matus.
Yeah, thank you. I'm going to ask if you have a significant number of reads mapped to multiple loci, is there anything you can do about it and use it, or it's just useless information?
So depending on what program you're using downstream, you can include it or not include it. And the program that we are going to use, we are throwing away that information and only using the uniquely mapped reads, which in most cases are sufficient. And I'm not going to actually not spend time on but yeah, if anyone wants to talk about this, that can be done in a private room. But basically what you can do is, of course, if you have one part of you have so let me see if I can paint this for you. You have 2 genes like this, and the part over here is the same. So basically when you map a read here, it's also going to map here, right? But then in this other part, they are unique. So so if you map a read here, it won't be mapped over here. And then what happens is that you are mapping multiple reads here. You're not mapping any reads here, and you're mapping multiple reads here. And then, of course, by looking at these reads that are here, they could either go to this gene or it could go to that gene. But since this other part of the gene is highly transcribed here, and this one is actually not transcribed here, can you actually, you have a eraser. Then these programs are sort of like, these reads are going to belong to this gene up here.
Okay, thank you.
So that's how they are doing it. Okay. Now I'm trying to find my pointer on the screen. You're wondering what I'm doing. Okay. Can we go on and take the other lecture directly? Is that fine? Give me a... Yeah, let's go.
Yes.
because yeah, so this is basically the last step. So now you've sort of made all the overview analysis, and now you basically want to go into the single reads, and you want to read the counts where are they, in which genes are they actually ending up? Is it gene A, gene B, or gene C? And this is a figure from feature count, where they are sort of describing the information on how do you assign a read to a gene. And by default, they are using this union. So that means that if you have a read and it overlaps the gene, it's going to be assigned to that gene. It doesn't have to, the entire read doesn't have to overlap the gene. It's still going to be assigned to that read. It can be that it's covering the part over the intron or something. It's still going to be assigned to that gene. If it has been correctly spliced, it will be counted as one count in that gene. If there are two overlapping genes in the annotation, if the part of the read is only in the fragment that is not overlapping, it's still going to be assigned to that gene. But as soon as it could either be attached to A or B, it's going to be say that it's ambiguous. So it's not going to assign it to any of the two genes. The only time it can assign here is if B is on the reverse strand and A is on the forward strand, and you have supplied stranded information of your read, because then it can say that it's either A or B. So this could be assigned to a gene if these two genes are on opposite strands and you have stranded information of the read. And this is also two other examples of when it becomes ambiguous. So there are different ways, so you start with a FASTQ file, and generally you can use two different programs to map reads to a reference. One is either by mapping it to the transcripts, or mapping it and then using this pseudo aligner, which I'm going to talk about on Friday. Or you do the way we did it yesterday, you map it to a reference. And then what we're going to do today is quantify gene levels using map reads and annotation. And the other thing that you were asking is this, where you quantify transcript levels. Yeah. Sorry. Yeah, and then in the end, you're ending up with either gene level counts or transcript level counts. And all the analysis that we are going to do during the rest of the week are gene level counts analysis. We're not going to focus on transcript level analysis. And the reason why it's another level of complication. And if you don't by forehand have a reason to think that there might be some transcripts, some isoforms that are more interesting than others, I and others most think that this extra step of complexity doesn't add anything. It just makes it more fuzzy. So the good thing is that the programs that are out there and all the different steps, or the ones that are sort of used, are all behaving quite well. But in this paper, they wanted to ask, do you get the same results when you're using qPCR as when you're doing this RNA sequencing analysis to get the counts from the reads? And the answer is yes. So what you have here is one sample, one sample, which is this MACA-CA, where they did qPCR on all the genes and got the counts for that. And then they've done different ways of getting gene counts, either by using a program called SAML or using TopHat cufflinks. So the TopHat is a RNA mapping program, or using TopHat with RSeQC. And this is as close as we get to what we are doing right now, or Callisto, which is the one that you're going to be trying out on Friday. And the level or the correlation between using qPCR and RNA-seq with all these different tools for getting gene counts are in the level of 85. Some are smaller, some are bigger, but yeah, in that order. And the ones that you don't get the same values on are the lowly expressed genes. And that information you can see in the bottom right corner here, where you have, sorry. Where you have expression on the x-axis. So the more highly expressed, the more to the right you are. And this is the distribution of expression of the genes. So it's this one here. And the ones in the other colors are the ones that are differentially expressed between the qPCR and the other samples. And the gray line, which is sort of this one, the one to the left here, that's the one that are differentially expressed between all the RNA sequencing with all the mapping procedures and the qPCR. So and this sort of, since you have the, I don't see my my own pointer. That's why so if you have, so you can see that it's shifted to the left. So that means that they are the lowly expressed genes. That's how you should interpret that one. If you look at the C, what you can see there is the ones that are differentially expressed between qPCR and all these different methods for assigning. And you can see that there are 182 that are found independently of what program that you're using. And then there are some that are unique depending on the method you're using. And the way I interpret this is that there are 182, or the ones that are in the bottom are the ones that are there because of there's a difference between qPCR and extracting reads from the sequencing machine. And the ones that are on the top, like 128 and 132, those are because of the different methods for mapping and extracting counts. So some difference between qPCR and RNA-seq is because it's two different methods for extracting. And then there are an additional layer because of the methods to determine what the read counts really are. The cool thing, which is comforting to know, is that is this. So let's see if I can clear this. Is that the differences that you have for extracting a number of counts per gene is very stable. So that means that when you're doing this into doing comparisons between samples, you are getting the same error for all the samples. So that means that when you start comparing between samples, even though the expression levels might be different between qPCR and the method, this method stays the same. So let me see if I can draw that, because this is kind of complicated, I guess. So the way I say it, at least. So you have the qPCR, and you have RNA-seq here, and you have gene A, and the concentration in this one in one sample is 1, and when you do the same analysis in using RNA-seq, it gets 10. So it should have been the same, but because of you're using 2 different methods, you're getting different values. But now you're taking another sample and you're measuring A again, and you see that this one now turns to 2. Then when you're doing this analysis in this method, it gets to be 20. So the difference between the samples is the same, even though there's an error in how highly expressed they are between the samples. So when you now start to comparing what are differentially expressed and how they are differentially expressed, and you look at that correlation instead, then you get these numbers. And then you see that there is almost perfect correlation. So independent on what method you're using, you're getting very similar results on what are differentially expressed.
Can I comment?
Yes.
Do they claim it for all of the methods for qPCR? Because it seems like now the golden standard for cDNA synthesis in qPCR is random hexamers with oligo-DTs. So that's the cDNA priming, where the priming for cDNA in RNA-seq is only poly-A.
Yeah.
So that's oligo-DT. And then you will get, as you said, well, like the absolute values are gonna be different, but then the transcripts, different transcripts are gonna show up also in qPCR, which cannot be seen in RNA-seq, which means that the transcripts which do not have poly-A tails.
Yeah.
So do they just comment on the specific poly-A tail cDNA synthesis, or is it?
It's a good question, and I have to read up on the paper again to answer that question. But it's true that, I mean, also in RNA sequencing, you can choose to do poly-A, or you can do ribo minus, where it's just a random priming, basically. And then, of course, you're going to be extracting different pools of the RNAs that you're looking at. So that's the good, and yeah, I think so. Then, of course, it really depends on what RNAs you want to look at, and so if you want to have, yeah. So that's where do I want to go with this? What I'm trying to say is that the values, or sort of the exact values or the expression levels of the genes are you should always take with some grain of salt. Like, is this value really true? Because of different, depending on what extraction method you used, what enrichment you did, and so on, you are going to end up with differences. The good thing is that most of these differences are stable. So if you do the same thing over and over again, you're getting the same differences all the time. And I think the most important thing when you're doing this is that you should try to keep the same protocols, both in terms of experimental protocols. So you extract the same thing out of your samples, but you should also use the same protocols when you're doing bioinformatics. So it doesn't mean, so in the end, when you start comparing counts, half of the library are poly enriched and half are not poly enriched by some other method, and half of them have been, you get the counts by using the way we did, and half of them are based on using pseudo align or something, because then you are starting to mix up everything and you get really strange results. So I think in general, when you're working with RNA sequencing data, by just looking at single samples, you can get some feeling for expression levels, but the real strength comes in when you start comparing samples, because then it's really stable. That was the long answer. Was that answering your question? Great. Okay, so what about if you want to go into transcript levels, what happens? Then there's this other paper that you can go in and read. I think there's a link to it somewhere, otherwise I will. Yeah, I think I showed it. It's in there. So and here they've done basically the same thing. You have FOSS-Q files. They used STAR as the mapper, and either they map to the genome, and then they used this path, which is cufflinks, to get the counts per transcript, or they've done this path by mapping to the transcriptome, just the transcriptome sequences, and then they have done all these different methods, or they've done pseudo counts by using these 3 different methods. And the take-home message is that all of them are doing a quite good job. So basically, the higher here, so in this case, they were mapping generated reads. So basically, they knew what the reads were coming from before, and then they were checking how many of these reads are actually going back to the place coming, or sort of assigned to the same transcript that they were coming from the start. And you can see that they are behaving equally well. And if there are any of the ones of these ones you shouldn't use, it's these cufflinks and express that seems to be behaving a bit worse. And the other thing that is also sort of a take-home message is that the more isoforms, so the more possibilities for a gene that are being given as a potential transcript, the more problem the program has to actually assign it to one of these isoforms. So if there are potentially over 15 isoforms annotated of that gene, the program has much higher problem to assign it back to the correct isoform. Whereas if it's not so many, so if it's between 1 and 5, it behaves much better. So this is, again, this of course makes sense, because if you have 15 or more different isoforms to put a read on it, you have to have a lot of information to sort of assign it to the correct one. The other thing that I just sort of want to mention is that then they started to compare between the different methods, how similar results are you getting. And in general, yeah, so these 3 here are these pseudo aligners, and there you get very good overlapping results, especially between Callisto and Salmon, which are the 2 most widely used. And this one is the first version, you could say, of these 3. So these 2 are behaving more similar. And then these are the 3 that where you were mapping to the transcript genome, and they are also getting very similar results. And then you have these 2 that weren't as good as, or didn't behave as good as the other one, and there you get more differences between these and the others. But independently, you can, I think all these scores are giving the information that you're getting approximately the same thing. It's really high correlation between all of these ones, I would say. So even if I would have been using Express or I would have found some things that I think are relevant, and I've used statistical tests to see that they are, and I found that they are conserved in other species, and it all makes sense. I wouldn't throw that results away. I would still use them because it's a good result there as well. Even though if I would have chosen now, I wouldn't have started with that one.